Hi, I'm Mushraq, and today I'll be presenting on behalf of myself and Dr. Jesse Boehm our progress on the Desmoid Tumor Dependency Map. The goal of the Cancer Dependency Map is to understand the relationship between the genetic alterations and molecular features of cancer and the dependencies they cause. We do this by systematically identifying genetic dependencies and drug sensitivities in expandable long-term cancer models. The goal is to discover targets and drug repurposing hypotheses and biomarkers that predict such dependencies. And we make all this information available to researchers on the web portal at depmap.org. So as a highly relevant example to our community, we can study the vulnerabilities associated with beta-catenin mutation status. And as a proof of concept analysis you see here, we can indeed identify expected targets. And so we hypothesized that mapping the dependencies of desmoid tumors will uncover beta-catenin-related as well as unrelated vulnerabilities. With increased number of cell lines for a particular T41 or S45 variant, we can also boost the power of detecting mutation-specific vulnerabilities. So thanks to the support of the Desmoid Tumor Foundation, we've embarked on a 3-year project to establish the first desmoid tumor dependency map. Our goal for this pilot study is to include at least 6 lines for screening, with the aim to understand the scale needed for identifying and validating vulnerabilities in this rare disease. And to do this, we need to turn early-stage cultures to long-term tumor-rich models so we can enable the CRISPR and drug screening. And in our first year, we're happy to report that we've successfully expanded and credentialed 2 cell lines that have now been submitted for genetic and drug screening at the dependency map team. We're also happy to report progress on our conversations with the NCI and ATCC to accept these lines that we generate as part of the HCMI project. And so now we can make the first desmoid tumor cell line publicly available at ATCC to be shared with researchers worldwide. However, our experience in the first year has also highlighted some challenges and lessons that have informed our strategy for the upcoming year. Thanks to the generous donation of cell lines from Drs. Raphael Pollock and Daniel Braggio from Ohio State University, we were able to track the expansion of a cohort of over 30 beta-catenin mutant desmoid tumor primary cultures using the single traditional media conditions we typically use, the DMEM plus 10% FBS. We conducted Sanger sequencing at every passage, and we identified cultures that maintained the tumor-rich content during the expansion process. From this, what we found reinforced the observations that I've shared in prior presentations that in most lines, the mutant allelic fraction falls below our detection limit. Specifically, we observed that the drop in detection success occurs earlier on in the expansion pipeline, suggesting that the non-mutant cells gain an early growth advantage when using this media condition. So we next turn to the experience of the Cancer Cell Line Factory group. And here, freshly received desmoid tumor samples were cultured in a combinatorial media screening strategy, and short-term cultures were analyzed by a beta-catenin ELISA as a surrogate for tumor cell content. A few selected cultures that meet the biomarker detection criteria and display high growth rate are then propagated further to establish long-term lines. From this, we find that at the earlier stage, multiple conditions attempted in every sample displays a tumor-rich readout on the biomarker assay. However, we also see a drop in mutant cell detection after long-term expansion, despite an improvement in the overall rate. This demonstrates that we can apply a media screening strategy to identify multiple conditions in which tumor-rich desmoid tumor models can be propagated for a longer period of time. And now this has motivated our next phase in the process, which is to apply a more sensitive method to quantify tumor purity across all conditions to improve the conditions we select for further expansion. So in order to meet our goal of studying the dependencies of more desmoid tumor samples, we need to improve our long-term model generation success. From the lessons we've learned from the 2 cohorts of samples just mentioned, We are currently embarking on an effort to systematically screen media conditions and track the allelic fraction as well. We think this will achieve two aims. One is to produce the additional cell lines needed for dependency screening. But secondly, our systematic screening will also help us uncover media conditions or additives that modulate the growth of the mutant or non-mutant populations. And this will help us explain why we observe the outgrowth of non-mutant cells with the current approaches. So how can we achieve the tracking of allelic fraction across many media conditions? Well, last year I proposed that we can potentially implement a bar-coded deep sequencing strategy so we can assess tumor cell fraction in a more sensitive manner and at a greater scale. Very briefly, every condition in our media screen would be provided with a unique barcode when amplifying the region of interest, and the products are then pooled across all conditions and across all passages and biological replicates. We then perform deep sequencing followed by a demultiplexing step to quantify the allelic fraction belonging to every condition. So today I'm happy to share the results from a pilot experiment. We pulled several admixed cultures in which we know what the mutant fraction is, and then we followed the proposed barcoding and demultiplexing workflow I just described to see if we can recapitulate the expected tumor fraction. The results of this experiment demonstrates that we can successfully compare the mutant cell fraction across multiple conditions of different degrees of tumor purity using our multiplexing approach. And as a further demonstration of the concept, we also followed our proposed workflow to quantify the tumor fraction in two media conditions known to have different effects on tumor purity in a melanoma disease case. Here, we show that our implementation helps identify a winning condition that maintains tumor purity over time. We also note that the differences between the two conditions could be identified as early as the first passage. So this would save wasted resources propagating normal outgrowths. So we are now excited to implement the systematic media screen as well as this deep sequencing strategy to potentially uncover similar trends in desmoid tumors. Optimizing and deploying our media screening and sequencing strategy is a priority for us this year because, in addition to producing more long-term models for screening, we think we can make significant contributions to understanding how desmoid tumor cells can be propagated and perturbed ex vivo. And we hope to share the resources we generate with the rest of our scientific community. We are also looking forward to evaluate the performance of the two cell lines we submitted for dependency screening, and the observations we'll be making from these two lines will greatly inform the scale we potentially need for generating the desmoid tumor dependency map successfully. We are also open to the possibility that not all samples are amenable for long-term propagation, but we still wish to include all samples we receive into our study. Importantly, we're clearly learning from this disease that the heterogeneity in cell composition is likely a feature and not a bug, and so we think that validating dependency hits in early admixed cultures will be key. Now, very briefly, I mentioned last year that we're also working on developing an image-based technique to help us study these tumors in their admixed nature. And this year, I'm happy to report some early demonstration for feasibility of this approach using gastroesophageal cancer samples as a proof of principle experiment. Very briefly, we find that the morphological features of the cell can be used to infer its current viability state as well as identifying its cell type. So I'm looking forward to further developing this method and implementing it for freshly acquired desmoid tumor samples because it'll enable subpopulation specific readouts at a much greater scale. Now finally, I'd like to thank the Desmoid Tumor Foundation for being the driving force of what started as an idea in this group and has now grown into the Rare Cancer Dependency Map. We recognize that many of the challenges and lessons we experienced are shared across rare diseases. And so I'm personally very happy that desmoid tumors is a flagship project within this effort. So I'd like to personally thank the Desmoid Tumor Foundation for their continued support of my research. And on behalf of Jesse and myself, I'd also like to thank all of our partners and collaborators, especially Barb and Irene with their RCRF, Dr. Pollack and Danielle Braggio from Ohio for their generous donation, and all of the Cancer Cell Line Factory team, especially Mooney, Rebecca, and Grace. Thank you very much.